One of the other things that we work a lot with here, in addition to cells, are actually flies. So flies are one of the traditional genetic workhorses of science. So this is, the green stuff at the bottom is the food, and what you do is you pick whichever strains of flies, mutant flies that you want, and you can actually mate them together to get specific genotypes. And so you put flies of different genotypes in the bottle, they will mate, and they will eat the food, and then what they do is, so the green stuff at the bottom is food, and there's all these little white things that are stuck to the side, and these are the larvae. So the larvae actually go down, eat through the bottom. That's why you can kind of see like a line through the bottom. It's kind of where the larvae have been eaten through, and then once they're ready to hatch, they climb up on the wall, and then they stay there, and then they transform, and they come out as flies. And so when this is a fairly large bottle, they're just keeping the stock going. So the dead bodies, depending on what you're doing, usually you end up sorting these. So flies are really interesting because we have all sorts of genetic markers for them. The genetic markers are like whether they have curly wings or serrated wings, or where they have white eyes, red eyes, different colored eyes. So you end up having to put carbon dioxide through a tube into here. They all fall asleep. You put them out on a mat that pumps out carbon dioxide, keeps them asleep, and you sort them under a microscope. And that's how you pick which ones you can mate in the next generation or do whatever your experiment is. And then you kind of just toss the rest of them. They get kept in the bottle and tossed.